Welcome back. We are going to be starting with the subsets of T helper cells. So various subsets of T helper cells exist. The type that develops during helper T cell differentiation depends on the type of antigen and the site at which it is encountered, as well as the cytokine exposure of the differentiating helper T cell. Helper T1 mediates most parts of the cellular immunity, including inflammation, macrophage activation, and differentiation of cytotoxic T cells. T helper 2 cells defend against parasitic worms by mobilizing eosinophils. They also activate responses dependent on humoral immunity, and they promote allergies. T helper 17 cells link together adaptive and innate immunity by releasing interleukin 17, which promotes inflammatory responses against extracellular microbes. T helper 17 may play a role in autoimmune diseases. The next type of specific effector T cell is the cytotoxic T cell. They directly attack and kill other cells. Activated cytotoxic T cells circulate in the blood and lymph and the lymphoid organs, looking for body cells displaying an antigen they recognize. Activated T cells target virus-infected cells, cells with intracellular bacteria or parasites, cancer cells, and foreign cells from transplants or transfusions. Cytotoxic T cells kill using two mechanisms. First, they release perforins and granzymes. Perforins create pores or perforate the cell through which granzymes can enter. Granzymes stimulate apoptosis. The second way is that cytotoxic T cells bind to a specific membrane receptor on the target cell, and that stimulates apoptosis, or cellular suicide. Figure 21.19a illustrates how cytotoxic T cells attack infected and cancerous cells. Cytotoxic T cells identify foreign antigens on MHC1 proteins and bind tightly to the target cell. Then, the cytotoxic T cell releases perforin and granzyme molecules. The perforin insert into the target cell membrane, polymerize, and form transmembrane pores. Granzymes enter the cell via the pores, and the granzymes activate the enzymes that trigger apoptosis. The cytotoxic T cell detaches and then searches for another prey. So we see all this occurring in here. Here's an SEM of a cytotoxic T cell killing a cancer cell. NK cells, which we discussed earlier, use the same mechanism to kill their target cells. NK cells, or natural killer cells, do not look for foreign antigens displayed on class 1 MHC proteins. Instead, they search for other abnormal signs like cells that lack the class 1 MHC protein, antibodies coating the target cell, different surface markers seen on stress cells. So, NK cells kill the abnormal cells that the cytotoxic T cells miss. We use the term immune surveillance to describe the process of the natural killer and the cytotoxic T cells prowling around the body looking for markers they each recognize. The next type of effector T cells are regulatory T cells, or Treg. Regulatory T cells dampen the immune response by direct contact or by secreting inhibitory cytokines like interleukin 10 and transforming growth factor beta. Regulatory T cells are important in preventing autoimmune reactions. They suppress the self-reactive lymphocytes in the outside lymphoid organs. Research is being done into using them to induce tolerance in transplanted tissues. Please make sure to study Figure 21.20. It is an excellent description of the adaptive defenses our body uses. The left side, shown here in the beigeish color, shows the cellular response. An antigen-infected body cell is engulfed by a dendritic cell. It becomes an antigen-presenting cell, presenting the self-antigen complex. This APC presenting the self-antigen activates naive CD8 T cells and naive CD4 T cells. The CD4 T cells are activated and clone into memory CD4 T cells and helper T cells. Helper T cells co-stimulate the naive CD8 T cells and give rise to memory and cytotoxic T cells. The cytokines helper T cells release stimulate nonspecific killer cells like macrophages and NK cells. Helper T cells also affect the humoral immunity seen on the right side of this figure. They stimulate antigen-activated B cells to clone and give rise to memory B cells and plasma cells. And we all know that plasma cells secrete antibodies. Table 21.8 is a good chart outlining the players in the adaptive response. So we see that B cells, plasma cells, T helper, cytotoxic T, regulatory, memory, and antigen presenting cell. All these, this is a great way just to kind of organize it in your brain. It also goes through antigens, antibody, perforin, granzymes, complement, and cytokines. All right, I'd like to play this movie. It's a short movie about the body's immune system, and it goes through all the parts that we've covered thus far. So sit back and enjoy.
Shown here in red, that enter her nasal passages and escape to the first-line innate defenses such as mucus and cilia. Some of these rhinoviruses enter the respiratory epithelial cells where they replicate, releasing more rhinoviruses into the patient's nasal passages and tissue fluids. In the tissue fluids, cells of the second line of innate defense, such as this dendritic cell, carry out surveillance for anything that appears foreign. Dendritic cells engulf the rhinovirus within an endosome. The endosome fuses with a lysosome, whose enzymes digest the rhinovirus. The resulting rhinovirus fragments are called epitopes. The dendritic cell then attaches the rhinovirus epitopes to presentation molecules called MHC2. This complex folds outward so that it sits on the outside of the dendritic cell membrane. The dendritic cell then enters a lymphatic vessel and travels to a nearby lymph node. Here, the dendritic cell, part of the innate defenses, encounters a helper T cell, which is part of the adaptive defenses. The T cell receptor, or TCR, of this helper T cell recognizes the rhinovirus epitope, while the CD4 glycoprotein associated with the TCR recognizes the MHC2. The dendritic cell then stimulates activation of this helper T cell. Adaptive defenses help to control recurrent infection, but also provide immunity against future infections by the same rhinovirus. They are carried out by two types of lymphocytes, B cells and T cells. Immature B cells conduct surveillance in the lymph nodes, using B cell receptors, or BCR, to recognize only one specific epitope. The BCR on the left does not recognize the rhinovirus, but the BCR on the right does, and this B cell is selected for activation in a step called clonal selection. The selected B cell takes in the rhinovirus, degrades it, and presents the rhinovirus epitope with its MHC2. Our mature helper T cell recognizes the rhinovirus epitope with its TCR and the MHC2 with its CD4. Then, the helper T cell secretes chemical messengers known as cytokines that activate the B cell. The B cell proliferates and differentiates in a process called clonal expansion, resulting in two types of cell. Memory B cells carry the memory of this pathogen for many years. Plasma cells secrete antibodies provides cell-mediated or cellular immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by its CD8 glycoprotein on the cytotoxic T cell. With the help of cytokines from a nearby healthy T cell, the cytotoxic T cell is activated and clonal expansion occurs, resulting in some long-lasting memory cells ready for a subsequent exposure, and many active cytotoxic T cells, or CTLs, that leave the patient's lymph vessel to search for infected cells that present rhinovirus epitope with their MHC1. The epitope is recognized by the TCR of the cytotoxic T cell, and the MHC1 is recognized by the T cell's CD8 glycoprotein. The cytotoxic T cell then secretes purpurine molecules, which form channels in the infected cell's membrane. Granzyme, also from the T cell, passes through the purpurine channels and activates enzymes that lead to apoptosis, the programmed death of the infected cell. The cytotoxic T cell then moves on to look for another infected cell. Cellular defenses are the body's big-time defense fighting the viruses and pathogens. Ultimately, when the antibodies conduct surveillance for rhinoviruses that are outside the body structure, in the patient's tissues and bloodstream, the cellular defenses, in the form of cytotoxic T cells, search for rhinovirus-infected cells, inducing apoptosis when they are found. Memory fatigue and T cells help our patient's body to recognize and more rapidly fight the sick rhinovirus vector death.
Okay, we're going to stop here for now.